We can think of high conservation value biodiversity, and this tends to be what both the public and I also think people in the oil palm industry exclusively concentrate on. So orangutans, rafflesia, gibbons, pitta here, a birdwing, and a cat, leopard. Now these are all very important, and really none of those is going to survive in an oil palm, more or less, and there's nothing you can do about this. These require forests, and you cannot conserve them in the oil palm matrix itself. But they're not the only animals there. There's also what I've called useful biodiversity, and that is those animals, and not just animals but plants as well, which contribute to ecosystem function. That is, they run ecosystems, and they're vital things for ecosystems to work. The ecosystem here, of course, is a plantation, which has a very specific purpose, and that's to produce oil palm, the highest possible yield. And these kinds of animals here are what we should be concerned about, as well as the other ones, as well as the kind of charismatic ones. We have here a range of soil invertebrates, a dung beetle rolling some dung, a predatory bug here which will help provide biological control possibly, a parasitoid wasp there, a fungus there which might provide biological control, a very famous example in oil palm of a barn owl eating rats, epiphytes which provide a lot of diversity in oil palm, and the pollinator. Now these animals, they're not just insects, but a lot of them are insects, are really important to the function of any ecosystem, any terrestrial ecosystem, including oil palm. And we must pay attention to them, I think, when we're managing oil palm. Now, I don't want to spend too long on this, because you all know it already. When forest is converted to oil palm, there is almost always a loss of both abundance and species richness of animals and plants. This graph shows quite a nice summary of that. In each case, the arrow in each case, the base shows the position when it was forest, and the tip shows what happens when you convert forest to oil palm. So, for example, this study done on beetles here started in the comparable forest area with that kind of abundance, x-axis, and richness of the y-axis, and after conversion to oil palm, it's moved down to there. So it's less abundance and less species richness. There are one or two small exceptions. The bees increase in species richness, surprisingly, when you convert a forest to oil palm. All of these are just one study. They could all be unrepresentative. I mean, they probably aren't, but you have to be very careful. They're not all show exactly the same thing. And it's very useful to have replicates. I'm going off to tangent here, but I think it's very often people think you've done one study on, say, ants in rainforest and oil palm, you don't need to do any more. Well, you do. You need to do as many as possible, because they could be unrepresentative or indeed wrong. Dung beetles increase in abundance in this study in oil palm. Mosquitoes, reduction in abundance, but the same number of species, and so on. But the general pattern, bats increase in abundance in oil palm. The general pattern is moving from high richness abundance to lower richness and abundance. Another point to make, which conservation scientists make, I'm not actually a conservation scientist, I'm an ecologist and entomologist. Conservation scientists make is that when you convert forest to oil palm, you must pay attention not just to the total number of species, but to what the species actually are. This is a study which we haven't published yet, done by Sarah Luke, working on the SAFE project in Sabah, and she is considering, amongst other things, dragonfly species richness in a range of forests. Now, if you look at this, the graph here on the right, you see these bars. On the left-hand side is the most oil palmy site, as it were, the worst site in terms of biodiversity, and on the right is Maliau forest, the best site in the area. Now, if you look at the colours on the bars, the blue ones show forest specialists. If you're a conservation scientist, this is what you care about. And you see that actually that doesn't mean an enormous reduction in species richness overall from the Maliau to the oil palm, but what has changed is the number of forest specialists. 100% of them in Maliau are forest specialists. The ones in oil palm, only a small number of species are forest specialists. The rest are generalists and so on. Things that don't matter so much in terms of conservation because they're not rare. And that's a fairly generally repeated pattern. The species change as you go from forest to oil palm, usually fewer of them, and almost always you're getting often tramp species or even pest species in oil palm, whereas in forest there are fewer of those, more specialists. Okay, to summarise that then, so high conservation value species, which the RSP I know are very concerned about. These are big organisms with a large range, and to conserve these, you just have to have large areas of forest. Fragments, as big a fragment as you can get, riparian strips and so on. And the big message here, which you all know, is that the high-yielding nature of oil palm makes this possible. That is, it is possible to spare land for nature because the oil palm itself is so high-yielding that you don't need an enormous area compared to soy or any other vegetable crop to produce the yield, and therefore potentially you can afford to let go some of the land in order to conserve these important charismatic animals. But I'm going to be concerned here really more with these ones on the right here, the useful species, small animals, which are vital to sustain ecosystem function. These things don't sustain ecosystem function. Orangutans and so on the whole, I mean, they do something. On the whole, because they're rare, they're not that important in terms of how the ecosystem works. That's a rough generalization of what these are. Okay, so this project is a collaborative research project between the Department of Zoology in Cambridge, where I am here, and SMART, Sinarmas, big company based mainly in Indonesia. And it's a large-scale experimental study of the relationship between habitat complexity, I'll explain this in more detail later, biodiversity, ecosystem function, and yield, the service, in the oil palm landscape. So the questions we're looking at is, it is a very simple model. Habitat complexity, that is how many understory plants there are, things on the epiphytes and so on, maybe riparian strips and so on, will have a positive effect. Increasing the complexity will tend to have a positive effect on biodiversity. Increased biodiversity will usually, but you can't assume this, you have to test it, improve ecosystem function, and improved ecosystem function will give you higher yield, possibly. So our questions are, our first question actually is what levels of biodiversity can oil palm plantations support? Now you might think we already know that, it's a rather boring question. I think we don't already know it, and it's not a boring question. People haven't actually really sat down in one sort of particular oil palm and looked at it for a long period of time to see what's there, across as many taxa as possible. And that's one of the things we're doing, and it's not that intellectually exciting perhaps, but I think it's really important. And we'll have some results I'll show you in a moment. And I think that will be useful for the oil palm industry, and to be able to say, and it's only one plantation of course, but what happens there, you'll be able to say at the end how diverse or how not diverse the landscape is, the oil palm landscape. Secondly, how does the link between these first three things, is there a link between habitat complexity, how many animal species there are, and how well the ecosystem function works? So the first bit there. And thirdly, and this is the question which I think in a way sort of ground truths the whole thing. There's no point having greater number of animals or habitat complexity if you reduce oil palm yield. If you reduce oil palm yield, then that means more land has to be given over to making oil palm, and the whole thing fails. Whatever you do here, I think, mustn't decrease yield. It must either have a neutral effect or increase yield, and we hope it will increase yield. Okay, where we are is in Sumatra. Unfortunately, we're not in Malaysia, but there we are. We're there in Sumatra, there near Pekanbaru, at the LIBO Smart Research Institute, which is there. This shows the various plantation sort of zones that we're working in. And this shows our experimental system. It's a relatively small area, but we chose this because the plots are fairly uniform. This just shows height, actually, so the pale green is the lowest. So all our sites are more or less on the pale or just in the dark green areas. And they're in triplets. There are 3 plots there. There are 6 on 1, 2, 3, 4, 5, 6. So that's where we're working, 18 plots altogether. And what we're doing is really quite a simple thing. We have 3 treatments in each of these triplets, arranged at random. So either, in the reduced one, in the normal treatment, we simply leave it as it is. In the reduced one, and it's 150 meters square with a 50-meter plot in the middle, the point of that is that means the plot in the middle is reasonably far from an edge, and you're really seeing what's happening within the plot. If you work on the edge, it could be that some kind of spillover to the edge. There's also a stream running along one edge of the plot. That's the ditch there, the blue thing. So the reduced thing is where we've done that now, removed all the ground vegetation by spray. The normal, if you just leave it as it is, that is normal management, the usual kind of control of epiphytes and the ground cover that Smarttree do. The enhanced one, we're doing something that no one ever does do on oil palm, well, unless they're irresponsible, I suppose, and that is just leave the ground cover to grow, leave all the understory to grow. And we are told that quite quickly trees and things will grow up under the oil palm. This is to give us really three extremes of habitat complexity. A very simple one here with no understory at all, and in some cases no epiphytes, a kind of normal one here, and the enhanced one there. And the whole point of this is to get some evidence that the understory levels are important and do something, and therefore, if we can find that, this will be good argument for stopping people being too keen to clear away the vegetation and make everything neat and tidy underneath oil palm, amongst other things. So, well, first of all, the first thing I wanted to do was to look at biodiversity in the plots. Now, in all these plots, although the sample size is quite small, we've got a before measure and a paired after measure in the same place. So one plot, we know what was there before, and we know what's there after the treatment. That's very important. That means we're not just comparing one area with another, it's the same area undergoing different treatments, longitudinal, time-based treatment. So I can't identify the plants at all, but these very impressive Indonesian people can, and I'm not meant to some of them, but they go around, it's me looking on, giving instruction there, and they identify the plants in the oil palm plot. There's an amazing diversity of plants. Many of you will know this within oil palm plantations. By the way, these are quite old plantations, they're about 20 years old, quite mature. Perhaps in that sense not typical of a lot of oil palm, but that's what we had available there. We're looking at insects. We're using a combination insect trap, which Ed Turner invented to use as safe, they're using it. It has an interception trap, so beetles that fly into there are knocked out, or knock themselves out and fall into a pitfall at the bottom. Or it's a kind of malaise trap as well. Smaller midges and things that fly in will go, and they fly upwards and into a bottle there and are captured there. And ants and things, there's a pitfall trap on the bottom, ants will walk into the pitfall trap. So it's quite a good way. It's kind of not a very good malaise trap, not a very good intercept trap, perhaps not a very good pitfall trap, but it does all three functions, and therefore it does quite well. We also canopy fogging, because there isn't really any other way of easily finding out what's in the canopy of the oil palm. So you just put the synthetic pyrethroid, wherever it is, into the canopy and have your tray underneath in the usual way and sample what's there. We're also doing kind of surveys of dragonflies, butterflies, assassin bugs, a well-known potential biological control agent, dung beetles and ants in specific ways within the plot. And we're doing that mainly by actually just walking around and identifying them by sight. We don't not kill, that's the dragonflies and butterflies, and assassin bugs. We're not killing them. We're also looking at vertebrates. Again, this work is done mainly not by us, but by the people at Smarttree. Rats in baited traps, camera traps for large mammals, and scats as well. Point counts for birds, Jake standing a point count there, and transects of frogs. We had a student from Princeton in America who did a master's looking at the frogs. That's another thing to say in general. All the time here we're looking for people to come in and work on these plots with their own expertise, their own group of animals, they're looking at all their own kind of function they want to study, and everyone's very welcome to come and help us. It's a very nice place to work, and there's a lot of support provided by Smarttree itself, the Research Institute. Now, that's the biodiversity. Now, we're looking at the ecosystem function as well, the kind of things that biodiversity is supporting. Looking at soil, physical and chemical properties. That's something which actually the oil palm company already does. I mean, I'm sure all oil palm companies do it, but that data is already there, and they're going to carry on collecting it, specifically in the areas that we're experimentally treating. We're looking at soil biological activity, use bait laminar strips there, which are kind of strips of plastic with holes in, and into that you put a particular material, and that's the speed with which that gets eaten away tells you how much general biological activity is going on in the soil. Measuring herbivory, simply to what extent the leaves get eaten by herbivores. And they chop a leaf off, is what's happening here, a particular age of leaf, and simply measure the number of fronds that are being eaten. Looking at leaf litter decomposition, again, this sounds easy, but anyone's ever done it. It's a terrible chore. I mean, you have to. So I say daily, if you go, you make sort of 3,000 little bags of leaves. So you make bags. You have to get the leaves. We use the oil palm itself, the oil palm frond. You have to get them dried, weigh them, put them into bags. We're using two sizes. That's the coarse one and the fine one, not shown here. Put them in the field, leave them for a certain amount of time, bring them back and weigh them. A very standard technique. In principle easy, but actually rather arduous to do. We're also looking at predation, and here, again, all the time we're trying to get a quantitative measure of whatever we're doing that's reasonably rigorous and reasonably representative of what's happening. So here we put tethered mealworms onto little bits of leaf and hang them up either free in the canopy or inside a cage. So this keeps volant, well, any kind of vertebrate out, rats, I suppose, and this is free to anything to eat. So birds and so on can eat what's on here. This could only be attacked basically by ants and other insects. So that gives us quite a nice idea of how much predation is happening. So the question here is, if you have all the ground cover taken away, and how much predation is going on? Are the predators still able to survive there? Or are they not there because there's no ferns and so on in the ground vegetation? We're also looking at dung decomposition, dung removal. This is a very, very important ecosystem function, not only because it removes dung, and for some purposes you have to use human dung, I'm afraid, but most of the time they use cow dung mixed up. So getting rid of the dung is really important, because if you don't do that, it can be generally unpleasant. But also, the beetles, of course, are aerating the soil. They're taking nutrients down into the soil. They're really important ecosystem engineers of the soil, as well as actually getting them dug out of the way. And this is Darren Mann here from Oxford, who's an expert on these with Eleanor Slade, and they've been doing a project kind of with us. And they're doing the dung beetle side of things. So again, it's very important to know, if you remove the understory or reduce it, does this affect the efficiency with which dung beetles do their business? We've also, the Princeton student called David Kurtz was looking at what frogs ate. Now, it's quite a good general thing to look at in an ecosystem. That is, how many species of frogs have you got, and how many species of ants or insects is each species eating? So is there a kind of very complex network, or is it very simple? It's a kind of one frog species eating everything, or 10 frog species, each of which eats lots of things. Now you can do this by a technique which I think the Germans invented, and that is you flush the stomach. It sounds cruel, and I think it can be cruel if you don't do it properly. So you have a syringe, and you simply syringe water into the frog's stomach, and it vomits. Well, not vomit exactly. It flushes out. Vomit is more kind of something happening from the animal. In this case, it's just sort of washed out into a sieve, and you collect what's there. And it's fairly easy to identify what they have eaten. I say fairly easy because sometimes the things have decomposed, but usually they haven't actually. The ants are still there. Insects are the cuticles are very tough, and you get a very good idea of what they're feeding on. Otherwise, it's quite hard actually to get an idea of the diet of any kind of group of animals in any ecosystem. This is one way of doing it. And of course, we're also at the end interested in the palm oil yield, which they're measuring anyway. We got to do it very specifically, tree by tree, in our area. Okay, so the treatment started, the initial spraying, on October 2012. We started getting the data on what animals and plants were in the plots in 2012. It's very important to have the pre-treatment data. And in February last year, the spraying started. And I must say, as a research worker, when you actually see being put into effect what you rather blithely thought straightforward, it was very grueling, really, because there were lots of these people with their very unpleasant herbicides, really, and going around spraying everything, and in black aprons, the massive effort, and a lot of unpleasant things brought on the environment, but we think it's a good experiment. And that's resulted, all that stuff has died now, and you see that's a reduced treatment plot there. Here's a normal one here. So the understory vegetation has gone. In this case, we haven't removed the epiphytes, but we are doing experiments on epiphytes as well. It's important to say you cannot do this kind of experiment unless you have full support of everyone working in the oil palm every day, more or less. Because people go around, not every day, but every few weeks, and check for regrowth and spray it again, or dig it out if it's growing. So it's kept free of weeds, it doesn't just happen once. This is our sort of, well, perhaps overcomplicated diagram of what we think is going to happen. So at the bottom, these are kind of things we're changing here. Basically, the understory habitat complexity, which will affect the microclimate, which we're measuring. It will then affect the diversity and abundance of lots of taxa. The plants themselves, of course, will be affected, and in general, they're not there. Predators and parasitoids will be affected. Rats, possibly. Decomposers, the pollinators, which we haven't started to measure yet. I'm not sure what we're predicting there. I think it's too small a scale, perhaps, to be very interesting, but since we're doing, we may as well measure them. Herbivores as well. We're also measuring ecosystem function in orange here, so predation, herbivory, nutrient cycling, decomposition, fruit damage, and eventually measuring yield. So we want to know how changes here, which is more or less the only bit you can control, might affect yield in the end. One side project a student from Copenhagen did was to look at this idea which you all know about, and that's beneficial plants. I was always myself been a bit sceptical about whether they do any good or not, and other people have done these experiments, but there's nothing really published that's very reliable, I don't think, or lots of things which suggest they're important. And she just made a start on this, and I think the evidence is quite good that these two, Turner and Antigonon, in some cases do have some effect on insect diversity, and possibly some effect by herbivory on yield, or we haven't measured yield itself. Now, so what results have we got? Well, for the dragonflies, we've now done quite a lot of sampling. We got 46 species from just the BEFTA plot, 16 quite small plots, 86 from the whole area of Smart Tree. This is reliable because we got someone called Rory Dow, who's this kind of world authority on dragonfly identification, because it's something an amateur can't do utterly reliably on their own. You do need someone who really knows, spent all their life collecting dragonflies, what he does. He's very determined. He gets his special dragonfly kit on, just kind of walks into the jungle, walks through rivers, and doesn't stop until it's evening. And then he's collected lots of dragonflies. 51 new records for the province of Riau in Sumatra, and 3 new records for Sumatra, just from our oil palm site. And that's really that tells you how little recorded dragonflies have been in Sumatra, basically. But even so, in an oil palm, it's not in forest, just in the oil palm itself, we've got 51 new records of dragonfly species which have not before been recorded in this part of Sumatra. There is a bit of an effect. These were too early into the experiment to give you any proper impressive results, but there does seem to be a slight effect here on the road sites. We looked in the middle of the forest, the middle of the plots, and next to the road and the river separately. There are far more dragonflies by the stream and by the road as you'd expect. There's more light there and there's water there. So these figures are all higher. But there is, even after only less than a year, a reduction in abundance of dragonflies in the reduced plots where there's no vegetation. This is not surprising, I don't think. I mean, there are almost no perching sites for the dragonflies and so on. So there is an effect there. As a kind of side project, we are making, wherever we can, identification guides and so on, so it's easier for people working in our park to perhaps do the identification of some of these groups. I mean, it's not that easy, and dragonflies are quite difficult, but with this guide, I think it should be possible to do it. Do a dragonfly walk and see what species you have. Here's Ed Turner, who did this work, showing these people how to identify the dragonflies. And that's the kind of key things we'll produce. But we have already produced that. Assassin bugs, there they are, very important. As you walk around, these things are very obvious. They kind of fly into you, and you can count them quite easily by walking, seeing how many you count. The treatments here are enhanced, that's where leaving the vegetation to grow, normal and reduced. And you can see in the reduced plot, that's the before and the after. So the pre-treatment is the first one, this is the post-treatment. Again, a very short time, but there's been quite a big effect. So certainly, they're both significant actually, in reduction and in the numbers. I'm not quite sure it's the numbers per kind of 10 meters of some area of walking. So if you take the vegetation away, you lose the assassin bugs, which is quite interesting, I think. We also, this is quite an aside actually, but a new species of Staphylinid beetle discovered from the oil palm plantation. I mean, you can discover new species quite easily if you're hard enough, almost anywhere, but it's quite nice to find that. This one here, rove beetle. Frog abundance, again, this was only 34 days after treatment, so I didn't really expect any effect at all, but there is some effect. This is before treatment here, the abundance. That's the control, not much change. And the reduced treatment, there is some suggestion that it's going down, and it's not meant from sort of an average of 13 species to an average of 8, which I think is quite surprising. So the frogs respond quite quickly to the fact that there's no vegetation, cover vegetation around them, and the species richness is also a bit lower. Just to comment on the dung beetles, what Eleanor Slade and Darren Mann found was, this is in relation to whether, it's a slightly different thing, which is not done in our plots, but in the same area of oil palm nearby. And they looked at the diversity and abundance of dung beetles in relation to whether or not there were cattle present. When cattle are not present, the abundance is a lot lower. So no cattle in these two estates here, a low abundance of dung beetles, higher with cattle. Biomass, as you'd expect, follows that. Species richness is not affected by the presence of cattle, just different species, small ones. The numbers of one particular species of very important dung beetle goes up dramatically in the presence of cows. Quite an interesting thing here is, where you don't have cows, the actual speed with which dung is removed, that's the ecosystem function you're measuring here, is much lower. It's not the, and the reason where there are cows, the dung removal, that's when you put experimental dung is removed very quickly. So presumably what's happening here is, because there are cows there, there's dung there, and therefore there are beetles there generally, whereas in the cow-free areas, there are no cows, no dung, and therefore no dung beetles. So any other dung that comes into this area here will not be removed very effectively, whereas here, because the cows are there kind of bumping up the beetle abundance, dung is removed very quickly. Litter loss, again, perhaps we have nothing to show here really, except it's working, and the different sizes of mesh affect the rate of mass loss, and it is being lost quite quickly, but we don't have comparisons between the different treatments. We've measured yield. Again, we don't expect to see anything yet because it's very early days, just to show, as you all know, it varies a lot seasonally. This shows the work done on beneficial planting, and one result. I've only got one to show you, I'm afraid, and that is actually with the Antigonon here, it's one on the left. If you remove, we did expect we'd simply remove the plant or have it there. When you remove it, the level of herbivory is affected. So with the beneficial plant there, it attracts in parasitoids and so on, and herbivory is reduced. Not only are there fewer things there, but there are more fewer herbivores, but there are more parasitoids and so on, wasps eating herbivores, fewer herbivores, and level of herbivory is reduced. So that's a start of some evidence that actually the beneficial plants may actually be beneficial. I think if they are, then I think the way they're used needs to be completely changed, and need to be not just in the thin strip where people can see them, which is what seems to happen now, but actually be planted where they might do some good. But it's early days. Okay, just conclusions then. I think this project, if it hasn't shown anything else, shows that I think the potential strength of research collaboration between industry and universities. It's been very, very helpful indeed. We can get access to all aspects of plantations management and help with logistics and with people. Very, very important to help us do our work. And the other results demonstrate, I think, high and variable and possibly valuable levels of biodiversity within plantations and potential impacts of understory management on biodiversity and on some ecosystem functions. Ed has done most of the work there, and there's the people who are working there to thank them. And that's it. Okay, I'm ready for questions now from the unseen audience. I hope you heard that. Not worrying. Okay, do questions. Who's looking to speak from the ether?
Do you have any evidence of differences in moth diversity?
Yes, I should have anticipated that. We haven't measured moth diversity. We almost certainly should have done, I think about it. It's now too late, of course, to go to the pre-treatment thing. But we can always compare the treatments now, the reduced, normal, and enhanced. We could put moths. I think the reason we didn't do it is that moth traps are rather uncertain distance in which they're actually gathering things. We could have looked perhaps at micro Lepidoptera actually in the leaves and things, but we haven't looked at moths.
Quite a lot of fungus feeders, fungus on beetles.
Yes, yes. I mean, is it known? Are moths diverse in oil palm? Has there people on the studied one? They're very low diversity.
Very low.
Yeah.
But it would be nice to have.
Yes. Yeah.
From the perspective of the plantation, what kind of result are they hoping to get?
I think, well, I'm not sure they're hoping for anything in particular. I think that what I'm hoping for, perhaps I can't really speak for them, is that we will show that the understory plants, the degree to which they're present, actually does affect both biodiversity and ecosystem function, and maybe yield. And that in itself, I think, is quite interesting. You can affect something by doing something quite simple. I'm not suggesting this is going to sort of change the whole science or anything. I mean, I think that it's just a simply one way of getting evidence for how diversity and ecosystem function could be linked. But there certainly is a lot of variety in the way in which the oil palm industry deals with the understory. When you drive around, I see lots of areas which have what in England you call a national trust approach, to kill everything, tidy. In other areas where some understory has been left. And my hunch, very strong hunch, is that is a lot better than removing it.
Well, what's the hunch? What's your expectations? I mean, the guess on yield. What do you think?
I have no idea. I wouldn't want to be tempted into a prediction there. I mean, no one anywhere except Taya Chantka looking at cocoa in Sulawesi or somewhere has ever shown, particularly, a link between biodiversity and yield of anything. It's something everyone assumes is happening in agricultural crops, and there's just no evidence. There's evidence that complexity affects diversity. It's a tiny amount of evidence that diversity affects function. To go right the way through to the crop yield. Not done really. So this is one. We'll provide one example. We hope of that.
Well, there is a correlation on yield and understory clearing.
Okay.
Mainly from the perspective of loose fruits. You're being able to see the loose fruits.
Yes.
Better.
Yeah. I mean, it certainly is the case that the control one isn't sort of vegetation everywhere. They're contrary to what people normally do. So there are paths, and it's just fairly easy to see things. And there's clear bit around the base of each tree, whatever they normally do, which won't be the same in different plantations.
Yeah, we have a question. The question is, can you describe the difference between full clearing and BAU? You mentioned that BAU included ground vegetation and epiphyte, clearing, which sounds the same as the clearing, which sounds the same as clear up.
So I'm not quite sure what BAU is.
Business as usual.
Sorry?
Business as usual.
Oh, gosh. Sorry not to be so incredibly ignorant. Can I get back to, how do I scroll back to that question? It sounds a very sensible question. Well, the difference is full clearing is killing absolutely everything, every scrap of vegetation. Not the epiphytes. I didn't describe that. We're doing the epiphytes separately in smaller areas where we're removing epiphytes by hand and seeing how that affects, how that interacts. If you remove the epiphytes when there's already nothing on the ground, or remove the epiphytes when the ground has got. So we're doing that separately. The difference is then, full clearing, you kill everything. Business as usual is a lot of the vegetation is left. In this particular plantation management anyway, I can see what the person's saying. It could be, if your plantation already killed everything, then there wouldn't be any difference between killing everything and business as usual, if business as usual involved complete destruction of the understory. But that's unusual, I think. Very. Hope that's answered.
Well, I have one question. Yeah. The three scenarios that you have, you don't have any control plots inside extra forests nearby.
No, you're good point. We have completely ignored natural forests in this, partly because there aren't any where we are. There's nothing at all. There's just oil palm. They have what they call their jungle reserve, but it's a very miserable thing, really. Not listening. It's quite small and very kind of trashed. And I also think it adds a fantastic extra level of complexity, and there aren't that many of us. That's really why we haven't done it. We're not worried about that. I mean, lots of other people have looked, and there are lots of groups looking at riparian strips and fragments. That's not what we're doing. To start with, anyway. Okay.
So you don't want to compare the foreign is found in plantation area compared to the natural areas.
People have done that to death. We know that already, I reckon. I don't see the point. I think people should stop comparing forest and oil palm. We've done it. And I think we should, I've got to contradict myself there, because I said at the beginning we need more study. But I think there's, I think that's not, given limited resources, the message there is fairly clear. I think we don't need to go on doing that. But I think we do need to look at, we very much need to look at fragment size, riparian strip width and so on, and distance from fragments and so on, rather than actually comparing just forest and oil palm all the time.
What about comparing small spots and...
Yeah. Yes, I think that's very important. We're not doing that, and I think someone should, as a separate study, look at smallholders and how. I don't know what their kind of policy, what the practice is to smallholders in terms of clearing understory and epiphytes and so on. I expect it's very variable. And Indonesia would perhaps be the place to do it, since there are a lot of smallholders there. I think for a foreigner to do, it's much more difficult for someone like me and my group to come in and start doing stuff with smallholders. I think local people should be doing that, because you need to talk to them, and we can't really do that. We'd have to do it via other people. And I think it's the kind of study which would be far better for. It's easier for foreign universities to work with oil palm companies, because they're companies and they do things. I think with the smallholders, I think it'd be much better for local people to be spearheading that myself. But it is a very good question. Things could be completely different. Yep.
Some of the biodiversity indicators that like frogs
yep.
and flies
aquatic local stage.
Yes.
So, with, for example, the quality of the water in the areas also play a other role compared to understory.
Yes, I think that's something we thought we were going to study in great detail because we deliberately chose this ditch along the edge of each of the plots. And we are looking at it, but the ditches are so fantastically variable that we've slightly lost heart. I mean, some they dry up a lot, and sometimes they're just not there. And it's just very hard to get a kind of satisfying, kind of rigorous comparison of the streams. Having said that, I have got a student working on the SAFE project in Borneo. I showed a slide, the dragonfly diversity was from her. And she's looking in great detail at how it's really riparian width strip, and also whether it's high forest quality or low forest quality next to the stream. How that affects the stream, lots of stuff on the kind of leaves and so on, and how that in turn affects the animals in the stream. So we're doing that, but not on this project. The frogs, well, yeah, I mean, there are streams going through these areas, and they are pretty dependent. I mean, they move quite a long way away from the streams, actually. And of course, the adult dragonflies fly a long way from the streams. But you're right, they do have an aquatic state, so it's very important. I mean, they have to be water there, or they won't be there at all. The streams are lovely here. I mean, I grew up with, not grew up, I mean, the first water I saw in oil palm was just awful. It's a terrible, I mean, sludgy, opaque, brown stuff. But these are clear, like limpid mountain streams almost. I don't quite know how they manage that. I think it's a very old plantation, I guess, they're eating. And that may be general in old plantations. But there's lots of stuff living there, lots of fish, which are not living there.
Any more questions? Dr. Foster?
How will I find something here? It's a mysterious machine. Patching.
Okay, not yet. Just okay, might be muted.
You are unmuted.
So thanks very much, Dr. Foster. Thanks for all those who are online watching this webinar. We hope to come back with more later on. Thanks for the questions.
Thank you.